Okay, hello everyone. So as I have promised you in the previous video, so let's continue with our story on nucleic acids, but this time I'm gonna take your time and I'm gonna tell you some of the basic information about RNA molecules. And let's start from the beginning, from what we have already learned from the previous chapter and slide. So we all know that DNA is a carrier, unique carrier of genetic information in a cell. And we all know from previous chapters, from the introduction to proteins, from DNA story, that actually the genetic information which is incorporated in the structure and sequence of nucleotides in DNA molecule is transcribed or transferred to the sequence of nucleotides in specific RNA molecules. And furthermore, RNA molecules are then actively involved in the transfer of those information to the sequence of amino acids in a future protein. So if you remember, we mentioned that proteins are tools of genetic information expression or instruments, and we have mentioned the very important role of RNA molecules in that transfer of hereditary information. So let's discuss a little bit about the primary and secondary structure of RNA molecules. So basically speaking, RNA structure is analogous to DNA structure. So we said that the DNA molecule is a polymer of purine and pyrimidine nucleotides, and we can also say the same for RNA molecules. So RNA molecules are polymers of purine and pyrimidine, but this time ribonucleotides. And like in DNA, and this time ribonucleotides are also attached to each other by the formation of 3', 5' phosphodiester bond. And this sequence of ribonucleotides in one polyribonucleotide chain actually describes the primary structure of RNA. So let's focus on the structure of each ribonucleotide. So each ribonucleotide consists of a nitrogen base, and nitrogen bases which are found in RNAs are purine bases, adenine and guanine, and pyrimidine bases, cytosine and uracil. Notice that here, instead of thymine from DNA, we have uracil, which is exclusive for RNA molecules. Then we have the sugar moiety, the sugar component, which is for RNA is strictly D-ribose. And you know that the presence of that OH group at 2' position of the ribose, if you remember the figure from the previous video. So that OH group actually enables the hydrolysis of RNA molecule or the degradation of nucleotides when that RNA is exposed to alkaline medium or when it's found in alkaline solution, which was not the case for DNA, if you remember. So also one of the important difference between DNA and RNA that RNA molecules are single-stranded molecules, but some shorter or longer double helixes can be formed in certain strand regions. Also, the parts of the strands are connected or stabilized by the formation of hydrogen bonds, and of course, again, following the base pair rule that we have already explained. So complementary, in those helicoidal regions, base pairs are attached to each other by the complementary rule or base pairing rule. So adenine with uracil here instead of thymine, and guanine with cytosine. The portion of helicoidal structure in different RNA types varies pretty much, but approximately it's 50%, ranging from a very high percent in tRNA, from a very low percent in messenger RNA, and so on and so on. And actually actually the presence of that smaller or higher portion of helicoidal structure is described by the secondary structure of RNA. Due to the fact that RNA is synthesized from DNA, and knowing the procedure of the RNA synthesis, we can conclude that RNA molecule is complementary only to one of the two DNA strands. So Chargaff rules cannot be applied to RNA as they could be applied to DNA, so the content of cytosine does not have to be equal to the content of guanine, and of course the content of adenine does not have to be equal to the content of uracil in the molecule of RNA. The other difference, one more difference, is that RNA molecules are predominantly much smaller than the DNA molecule, which is logical because RNA molecules are actually obtained as copied fragments of the DNA molecule. So they can be smaller or larger depending on the type of the RNA and depending on its future role in the protein synthesis. So basically, there are three types of ribonucleic acids: ribosomal RNA, marked as rRNA abbreviated, then transport RNA, tRNA, and messenger mRNA, and they are directly involved in the protein synthesis. Speaking of the distribution of RNA molecules in a cell, approximately 11% is found in nucleus, approximately 15% in mitochondria, 24%, like 1/4 of the total amount, in cytosol, and over 50% in the ribosome, which is again pretty logic because the protein synthesis occurs in the cytosol and it's located on ribosomes, so obviously the concentration of RNAs will be on ribosomes. So there are also some other RNAs which are definitely less abundant. Those are minor types of RNAs, and they can be involved, for example, in the processes of replication. So you will learn that for the synthesis of lagging strand, you need those primers which are going to be initiate the synthesis of Okazaki fragments, and they're of the RNA nature and so on. Or they can be involved in the modification of RNAs to their final form. But the quantity, the total amount of RNA molecules, it's much higher than the content of DNA, because there is the need for the synthesis of a lot of types of RNAs which are going to be involved in the synthesis of different proteins. So 5 to 10 times more RNAs are found than DNAs. Also, some of the RNA molecules can act as catalyzers as well, or catalysts. So in the action of ribonucleases, which are involved in the degradation of other RNA molecules, you have the catalytic function of RNA fragments. And in RNA polymerases, so they catalyze the formation of phosphodiester bonds. We can also illustrate the involvement of RNA molecules in and their catalytic function. So there are some differences between three major classes of RNA molecules, and they differ according to their base content, of course, which is logical, according to their secondary structure, and we will discuss about it in the upcoming slides. About their size, the smallest are tRNA molecules. Their variety, our RNAs are pretty unique. There are just several types of our RNA, and there are slightly more types of tRNA, for example, like 60, and there are a huge number of different mRNAs. And they also, of course, differ upon their biological function. So, as I have mentioned, tRNAs are the smallest, and they contain approximately 80 nucleotides or 80 base pairs, and their size is marked with the 4 Swedberg units. I'm going to get to those Swedberg units in the next slide. So rRNA in prokaryotes, they contain approximately 1,600 nucleotides. You don't have to remember those nucleoside types. Just remember the pattern that tRNAs are the smallest, and 3,200 nucleotides, while eukaryotic cells, they form 2,050 nucleotides with 18 Swedberg constant units and 5,500 nucleotides with 28, the huge 28S units molecules. So one type of RNA, they can have the same primary structure, but tRNAs differ due to the specificity for certain amino acids. Also, speaking of differences in the base content, during the evolution, the development of rRNA and tRNAs went in the direction that they contain more guanine-cytosine base pairs. So they have more pronounced secondary structure. So you will see some coils and some helicoidal structure in those two types of RNA, and some of them are pretty metabolically stable, especially tRNA. On the other side, messenger RNAs are very heterogeneous, because if there are so many different types of proteins in our cells and body fluids, there are going to be that many messenger RNAs coding the amino acid sequences. So in terms of the base content and size, of course, they're also they can be small if they're coding the biosynthesis of a smaller peptides containing like a couple of dozens or a couple of amino acids in a sequence, or they can be huge if they're coding proteins containing several thousands of amino acids. So it depends on what they are coding for. But all of those three types of RNAs are synthesized in the process of transcription, and you're going to go into details about this process later on in some of the upcoming biochem chapters. So let's start from the beginning. So we have ribosomal RNA, rRNA. So its name says that they form ribosomes. So rRNA actually is found in approximately 65% of ribosomes. And ribosomes are nothing else but some subcellular ribonucleoprotein complexes. And they're actually the places at which protein synthesis occurs. So they're considered to be like, like fabrics for the biosynthesis of proteins. And rRNA is the largest group of RNAs, so approximately 80%, but it has just a few specific types. So there are a lot of RNAs, speaking of the portion of the RNA, rRNA in the total amount of RNA in the cell, but just a few specific types. So in prokaryotes we have 3 types and 4 types in eukaryotic cells, and they are, as I have mentioned, marked by their Swedberg units or sedimentation constants. Just to explain to you, if you still don't know about these S units, so they refer to the sedimentation coefficient of macromolecules, and actually as Swedberg units, they represent the unit which is proportional to the sedimentation rate in a density gradient. So it's taken as a unit of a molecule size. So the higher the S, the larger the molecule. So in prokaryotes, you have like only 3 different types of rRNA forming 2 subunits of ribosome. So if you take a look at this slide, so one prokaryotic ribosome is 70S, of the size 70S, and it contains the large subunit and the small subunit. The large one is 50S, the smaller one is 30S. and the large subunit contains 23S and 5S rRNA and approximately 30 different proteins, and this is summarized here. And the small subunit contains only 16S rRNA and approximately the total amount of 20 different proteins. When those two subunits are condensed, they tend to form the functional ribosome, which is involved in the protein synthesis. In the figure below, and it's summarized here, in eukaryotic cells, a ribosome is of 80S, so it's slightly larger than the prokaryotic ribosome, and again it constitutes of a large and small subunit of the ribosome. So you have the large subunit containing 28S together with 5.8S here attached and 5S rRNA and a total amount of approximately 50 proteins. And the smaller subunit of 40S, it contains 18S rRNA and approximately 30 different types of proteins. Usually students ask me if they have to remember all those S units and S sizes. Yes, because we're gonna get back to this again when during the chapter on protein synthesis and translation. So we will have to explain which are the roles of these specific rRNAs and which are the roles of those subunits in the protein synthesis. So yes, please start memorizing now, and you will feel better when we move on to translation when knowing these values at this specific moment. So this is the summary of the figure represented earlier. So let's move on to transport RNA. So those are, as I have mentioned, relatively small molecules containing not more than 90 nucleotides, so approximately 80 nucleotides in a sequence. We said that there are only few types, like 3 in prokaryotic cells and 4 in eukaryotic cells for rRNA, but now we have a slightly more different types of tRNAs, approximately 60. And what is the rule? Each amino acid to be of those 20 proteinogenic amino acid is attributed at least one corresponding tRNA, but some amino acids may be attributed more tRNA, so more than one. And for example, there are 5 different tRNAs for carrying the amino acid leucine. So in the structure of tRNA, in the sequence of nucleotides in tRNA, apart from those 4 common bases, adenine, guanine, cytosine, and uracil, a tRNA contains more than 10% of so-called rare or unusual bases like thiouracil, like acetylcytosine, and so on and so on. Also, tRNA has a very specific 3-dimensional structure because more than 70% of the sequence is actually in the form of the helix, and it forms like 3 loops, of which one contains a very specific base triplet called anticodon, and it's complementary to the adequate codon on mRNA. So here is the figure representing those 3 loops, and one of those loops contains 3 bases marked as anticodon. And of course, there is in some types of tRNAs there is one extra arm which is not found in all tRNAs. So this can be a wobble base, but we will discuss it in some further chapters. And what else should we notice here and emphasize here? At the 3' end, take a look at the figure. tRNAs, all tRNAs have the same triplet of bases in the sequence CCA, where A, adenylic acid, is the last and at the very end, and it has 3' OH group to which an adequate amino acid can be attached by the formation of ester bond thus forming activated amino acid or amino acid tRNA. On the other side, and a lot of students actually somehow forget this information, which is of crucial importance for understanding the protein synthesis. So I'm going to repeat it again. At the 3' end, all tRNAs have the sequence CCA, where adenylic acid or adenylate is the last nucleotide with the free OH group at 3' position, which is ready to react with a carboxyl group of an amino acid, thus forming an ester bond and activating amino acid and getting it ready for the protein synthesis and forming a so-called amino acid tRNA. On the other side of the chain, or at the 5' end, guanylic acid is usually found, or guanylate, and it's usually phosphorylated. So what is the shape of the tRNA? It's a very specific, so according to this scheme, it's in the form of a kind of clove leaf. The activation and transfer of amino acid to ribosomes, where protein synthesis take place, and actually the pairing of those 3 bases of the amino acid tRNA, anticodon, is paired with 3 bases of the codon in the coding region of mRNA, and of course following the base pairing rule. And finally the last one, the last but not the least, that is the messenger RNA. It contains, there are really thousands of different mRNAs. Depending on the structure of the protein they're actually coding, and there are some specificities of eukaryotic messenger RNA molecules. So they have a long sequence of adenylic acid at the 3' end, and it's called poly-A tail, which you can see here from the slide. And also at the 5' end it has a cap, so-called cap. So it's composed of 7-methylguanosine, which is attached via 3 molecules of phosphoric acid, if you can also notice from the figure here. So the messenger RNAs are synthesized in nucleus like all other types of RNAs, and they are nothing else but complementary copies of DNA. Then they are transported through nuclear membrane and to cytosol and then to ribosomes, and they are going to serve as unique templates for protein synthesis. So messenger RNA is nothing else but a messengers of the information which has to be transferred from DNA to proteins. So each protein has its specific messenger RNA, or at least its specific part. In each RNA we have a very specific part which is marked by the triplet of bases, so 3 bases in a row, marked as codon. In prokaryotic cells, one mRNA codes 1 or more proteins, so it can be monocystronic or polycystronic, rather polycystronic. But in eukaryotic cells, one mRNA codes exclusively only one polypeptide and protein, so it's exclusively monocystronic. The other difference between prokaryotic and eukaryotic cells is that mRNA in prokaryotic cells is absolutely functional immediately upon its synthesis. So it's considered to be mature mRNA in the way it's synthesized from the DNA template. But in mammals and humans, usually the synthesized mRNA is just the primary transcript or the primary product, and usually it's called heterogeneous nuclear RNA. So it's just the beginning, it's just an initial molecule which is not functional, it cannot be involved in protein synthesis, but it has to undergo certain modifications to become mature and to become functional. So heterogeneous nuclear RNA actually contains regions which do not carry the information about the amino acids to be incorporated in the structure of a protein. So we call those regions non-coding regions or non-coding nucleotide sequences. So those coding regions are marked as exons and those non-coding regions are marked as introns. So if we want to activate, let's say, messenger RNA, if we want to make it mature, we have to remove introns from the sequence and to put those exons together by the procedure of splicing. L is missing. I am so sorry for my typing mistake. SPL, splicing. Usually those introns are of the very specific structure and nucleotide sequence, so they usually start with the GU pair and they end up with the AG pair, but before that last AG pair they have pyrimid enriched sequence, and those specific domains of introns are actually signals to specific enzymes to cut them off and to mark them as non-coding regions, and then to put together and by ligases the coding exons. And finally, when we have removed the introns, when we got our exons back together, mRNA gets a cap and the 5' end and the poly A tail and the 3' end. So finally, they only approximately 5% of the total age-end RNA is transformed to functionally mature mRNA. And then mRNA actually consists of those triplets of bases, which are marked as codons, and they are carrying the information on the amino acids to be incorporated in a future protein. That bunch of those codons is marked as genetic code. And I'm going to tell you more details about the genetic code and its transfer to amino acid sequence later on when we're about to discuss the protein synthesis or translation process. But until then, that was more or less all that you need to know about the specificities of RNA molecules, types of RNA molecules and their function. So until some other topics, until the story on some other biomolecules, maybe lipids or carbs, stay safe, have a nice rest of the day, and see you soon.